Hi everyone. We're going to go through the transcription lecture today. Before we get started on transcription, I want to bring up a couple of things just to show you what you might need to go through today's lecture. So the first thing is on our course Blackboard page. Here is our course homepage right here, course information. You'll notice the revised syllabus, which will tell you what we're doing each day in class, and today's lecture is transcription. Fridays will be translation, and I'll get that ready for you as soon as I can as well. And so if you want access to the PowerPoint slides themselves so you can look through them as slowly as you want to, so that you can look through the notes, you're just going to go to Lecture Materials, go down to Unit 3, and then in the lectures, you'll click on Lecture 3, and you have the PowerPoint and the PDF available. And remember that in the PowerPoint slide, there are often some extra notes in the notes section, which will have definitions of some of the key words that are in bold, or in this lecture, some of the keywords are in red boxes. So make sure you check out all of the resources that are on Blackboard. Have the PowerPoint ready. And then also, we've got our course pack right here. All right, so before we left for spring break, we started transcription and translation. So this is lecture 3 within unit 3. We've done the introduction already, talked about the general processes of transcription and translation. We talked about the 3 different types of RNA, where these transcription and translation happen in prokaryotes versus eukaryotes. And then we covered an introduction to the genetic code. All right, so when we get to transcription, a closer look, here's where we're starting off. And in your textbook, that's concept 17.2. So I would go ahead and do the reading for concept 17.2 and jot down or note any questions that you have. And you can use the discussion board that I set up to do that as well. All right. And so here is a picture of transcription right here. I'm going to draw a little bit of this in your course pack. So over here, here's our course pack. Oh, in general, what we've got going on with transcription is if we were to write down in simply what it is, we would say transcription is when RNA is created or synthesized from a DNA template. So that's the general description. Another way that I sometimes will put it is we have DNA, and from that DNA, we're making RNA. Okay, so we're going to start with our DNA. So we've got 2 strands of DNA. One of the strands is going to go from 5' to 3'. And then the other strand here is going to go from 3' to 5'. Now RNA is going to be made by an enzyme called RNA polymerase in the 5' to 3' direction. So that means the complementary strand, or the one that goes from 3' to 5', this one is going to be the template strand because it's going to be complementary to the RNA that's going from 5' to 3'. So the template strand is the strand of DNA that's going to be transcribed. So the template strand, this is going to be the strand that we are transcribing. So if you are ever asked to transcribe something, you're going to start with DNA, and you're going to start with that template strand that goes from 3' to 5', and you're going to use RNA nucleotides that are complementary to the DNA nucleotides. All right, so this is an overview of what we're going to cover. And so going back to the initiation of transcription, which comes first, we've got some of these key words that are in boxes. And so the promoter region is where transcription begins. So to put it simply, this promoter region right here, you can write this in your definitions in your course pack, the promoter region is going to be where transcription is initiated, or where it begins. And within this promoter region, it's just a sequence on the double-stranded DNA. And there's a series of letters at the beginning of the promoter, a string of A's and T's, and so we call that the ta-ta box. So at the promoter region, we're going to get transcription factors. These are proteins that bind at the promoter. And what they do is they recruit RNA polymerase to come to that site. So these are proteins that bind at the promoter. And here you see RNA polymerase joining, and the RNA polymerase only comes when the transcription factors are bound. So we say that the transcription factors recruit RNA polymerase. So once they're there, RNAP, or RNA polymerase for short, will bind at the promoter. Now, RNA polymerase is the important enzyme in transcription. It's going to do two things. Once it's bound at the promoter, it's going to start to unwind the double-stranded DNA. And that way it has access to the template strand so it can start building the RNA transcript. When all of these things are together, we call that the transcription initiation complex. So the transcription complex is when RNAP is bound at the promoter. Or to the transcription factors at the promoter region. Okay, so going back to our picture here, the promoter region is where transcription begins. I'm just going to kind of draw that here. At the very beginning of the promoter, we have something called the ta-ta box. All right, so that's a string of A's and T's right here. And overall, we call this region the promoter. So this is where RNA polymerase is going to bind. So the transcription factors bind first. That's a really bad drawing. And then comes in RNA polymerase. Okay, so RNA polymerase, I'm going to switch colors here so that we have a different color for RNA. The next thing that happens after RNA polymerase binds, transcription has been initiated, and then all it has to do is proceed from here. And so RNA polymerase is going to move in this direction along the template strand, colorful. And as it does that, it's going to create a messenger RNA strand that runs from 5' to 3'. All right, so here is the transcript that is made, and the transcript is the mRNA. Ooh. Nope, I didn't undo. Here we go. The transcript is the mRNA that's made from transcription, or just the RNA that's made from transcription, because other types of RNA can be made as well. Okay. And so RNA polymerase, the second job, I said it has one job. The first job was to separate the double-stranded DNA. The second job of RNAP is it's an enzyme that creates a polymer of RNA. So it synthesizes the new RNA transcript. Synthesizes the new RNA transcript. And this transcript, again, is going to be complementary to the template strand. Okay, I'm going to go back to a different color here. It does this until it hits a termination sequence. And so the termination sequence, use blue. All right, the termination sequence is going to be along the DNA strand right here. Right, we're going to call this the termination sequence. This is going to be where transcription ends. Okay, I'm going to show you here in your book diagram right here. The termination sequence is going to be a sequence that signals the formation of a string of A's on the transcript. So the termination sequence is often called a termination signal, and it'll signal for the formation of a 6-letter sequence AAU, AAA at the end of the messenger RNA transcript. When this is formed, the transcript gets cleaved from the RNA polymerase and transcription ends. RNA polymerase will fall off, and now we've got our completed transcript. Right, so in prokaryotes, that's that. In a prokaryotic cell, this completed RNA transcript will immediately get read by the ribosome in translation. But in eukaryotes, this process right here is happening in the nucleus. And the RNA transcript has to go from the nucleus out into the cytoplasm before a ribosome clamps on. And this process in a cell takes, I don't want to say a long time, it takes less than a second, but during that time, enzymes will start to degrade the RNA. So the RNA is going to be processed in eukaryotes to help it stabilize on that journey from the nucleus into the cytoplasm. And so in our next slide, in post-transcriptional modification, this is going to happen in eukaryotes only. Okay, so in eukaryotes only, and that is because only in eukaryotes the RNA transcript has to travel from the nucleus into the cytoplasm. Through the nuclear pores. All right, and in prokaryotes that doesn't happen. And so there are two things that will happen to the transcript. The first thing that will happen is the addition of a 5' cap right here. This 5' cap is a modified guanine nucleotide that is added to the 5' end of the transcript. Or in eukaryotes, we'll call it a pre-transcript. The second thing that happens is the addition of a poly-A tail. And this poly-A tail is the addition of several more adenines that are enzymatically added to the end of that RNA transcript at the very end of transcription. All right, so these two steps right here, these are processing steps or steps that happen after transcription, so we call it post-transcriptional modifications. And these protect the ends of the RNA strand so they get less degraded. So the ends are protected through these two means right here. And then the second thing that happens is that the introns are cut out or spliced out. So when introns are removed, we call that splicing or spliced out. All right, the introns are the segments that are going to be removed. And the exons, the way that I remember this, is the exons are going to be the ones that are expressed, or the ones that are eventually going to code for proteins. So we've got the 5' cap and the poly-A tail, and now we have only our exons left after the introns have been spliced out, or the exons are spliced together. So this right here we call the pre-mRNA, in eukaryotes. And then once these modifications happen, now we have our RNA transcript. This transcript will go through the nuclear pores out into the cytoplasm, and now it's ready to be clamped on by a ribosome and read for translation. So that's the end of transcription and post-transcriptional modification. What I want to do quickly is bring up your study guide. So we went through transcription. We talked about the initiation of transcription with these 5 terms. The transcript is what's made from transcription, and the termination sequence is where transcription ends. We also talked about why post-transcriptional modifications or RNA processing steps only happen in eukaryotes. And we talked about the 5' cap and the poly A to tail being added to the 5' and 3' ends of the transcript, and this protects the ends from degradation. We also talked about how the introns are cut out or spliced out. Excision means to cut out, and that's called RNA splicing. So the exons are the segments that are expressed and the ones that are kept and spliced together. So we're going to move on to translation in our next lecture. But after this one, make sure you go through the terms that related to transcription, ending with exons right here. And in our concepts, you should know how to transcribe a DNA strand, that the template strand that goes from 3' to 5' is the one that is transcribed. And you should also know the major players involved in transcription, the enzymes and transcription factors, their functions of each of the bold words that we put in a red box or we had in bold in your course pack. And you should be able to identify where it's initiated, how it's carried out, and how this transcription process is terminated. You should also be able to cover what those post-transcriptional processing steps are and describe why they occur in eukaryotes but not in prokaryotes. So that ends the lecture on transcription. Next up will be translation. Thank you.